Good evening, everyone. Thank you for joining us tonight for this final event of our RiverWatch Fall Speaker Series. For those of you who are not very familiar with Illinois RiverWatch, we are a community science organization that trains volunteers to monitor water quality in their own communities. The mission of RiverWatch is to safeguard the future of Illinois rivers and streams through stewardship, education, and sound science. So tonight, our presenter is Brian Saccardi. As an environmental scientist, Brian studies the critical zone, or the surface of the earth encompassing the forest canopy to the groundwater and everything in between. Most of his current research focuses on carbon cycling and the processes that transport carbon through our soils and streams. Brian studied wildlife conservation at UConn, soil and water management at the University of New Hampshire, and the critical zone at UMass, where he eventually got his PhD in geoscience. Along the way, he worked for several environmental organizations, including Great Mountain Forest, Housatonic River Valley Association, and Sessions Woods Wildlife Management Area. Brian, thank you so much for being with us tonight, and I will hand things over to you.
Cool. Yeah, happy to be here. Well, thank you for that introduction. Um, as mentioned, I'm Brian, and I'll be talking today about some of my research with the CZNet or CINet cluster at the University of Illinois, specifically on carbon cycling in agricultural and prairie soils of the Midwest. Um, so to kind of get started, I'll be doing a bit of just a bit of a walkthrough on what we're going to cover today. I'll start with some background about what the critical zone is, what soils are and why they're important, a bit about the carbon cycle, and then some feedbacks, in order to kind of give some background about what my research is and why I think it's important and how it fits into kind of broader scientific topics. And then we'll go into my research, the methods I use, what my findings are, and a few conclusions. And so to start off, what is the critical zone? Kind of in the introduction there was a really good example or definition of the critical zone. It's from the bedrock to the tops of the trees. It includes the root zone. It includes the plants, near surface environment, river corridor, all of that stuff. And often I hear it referred to as the thin film of life that covers the Earth's surface. And so that's kind of generally how I like to think of the critical zone is that region of Earth that has the abundance of life. If you want a more scientific definition of it, it's the shallow subsurface, surface, and lower atmosphere of the terrestrial environment. Um, so once again, the bedrock to the tops of the trees, that layer of life on Earth. And so that's what the critical zone is, but why is the critical zone actually important? Um, and it's important for many reasons. Any environmental reason that you can think of, and many of our economic and other needs as well are supplied by the critical zone. So rivers supply drinking water, energy, habitat for wildlife, recreational options. Soils are what we use for food and agriculture. They provide forests, additional habitat. They store carbon and groundwater. And then the atmosphere regulates temperature, carbon cycle, provides oxygen. And so the critical zone is the layer of Earth that supports life, and therefore it is very important to understand it in order to understand what these resources are and kind of how they help to support life on Earth. And so if you think about it, though, a lot of what I've mentioned um, is studied in other forms, right? We have soil scientists, we have hydrologists that study rivers and streams and lakes. Um, we have atmospheric scientists, but the focus of the critical zone is more on a holistic view of the system. We study the connections. We study how the terrestrial environment um, affects rivers, such as how nutrients flow from soils into rivers to the oceans. We look at how soils can be a sink of carbon for the atmosphere. Um, and so we look at a lot of the connections that go along kind of between these more regular boundaries of different scientific disciplines. And that's kind of the focus of a lot of critical zone research is how these different parts of our environment interact with each other. And so that is kind of a tall order, right? It's hard to do everything all at once. So we even still have to pick and choose connections that we want to study or that we want to focus on. So for this research, I've kind of chosen the soil environment and specifically looking at prairie versus agricultural soil environments. And then my interest is really in that rooting zone and with CO2 respiration and carbon cycling itself. And so, although we look at the system kind of as a whole and those connections, we do have to kind of tease down to bite-sized chunks that we can understand and research at reasonable timescales. So that's a bit of a background of the critical zone. Next, we'll step into a bit about soils. And so, first off, what are soils? Like, what are they made of? In general, soils are a conglomeration of gases, water, minerals, and organic matter. This bottom left-hand figure here is a pretty good generalization of soils, right? 25% water, 25% air, 45% minerals, and about 5% organic matter. These numbers are a good generalization, but they're not necessarily true for all soils, especially all times too. So, like directly after a rain event, your soil will have probably a lot more water and a lot less air. If it's been dry for a while, that water may have evaporated or transpired, and the air content of the water will go up, or of the soil will go up, whereas water will go down. Also, for the mineral versus organic matter percent, this is, as I said, a good rule of thumb, but if you're in a bog or a wetland or even a good garden soil, the organic matter will be much higher than this. So this is a good generalization, but soils are different depending on the environment that kind of produced them. But there's more than just those physical aspects of the soil. There's also the biological aspects of soil. And soil also then consists of roots, fungus, and microbes and bacteria that kind of play a key role in the chemical and biological processes of what's going on in that soil itself, as well as kind of maintaining soil structure. And so that's what soils are made of. And kind of following the same theme, why are they important? Why do we kind of care about soils in general, and why study them? There's a lot of reasons. So soils help to cycle nutrients for plants, trees, and other kind of organisms in general. They store water and regulate water release. They sequester carbon as well as other greenhouse gases. Um, they can regulate water chemistry, and they're a habitat for a lot of different microbial and animal communities as well. Additionally, they are used, kind of if you look down at this bottom right-hand corner of this figure here, as an engineering medium for roads and houses in our built environments as well, outside of the natural benefits that they give us. So, kind of to go through to the next kind of background piece, the carbon cycle. And so what is the carbon cycle is once again kind of to follow the trend, our first question. And the carbon cycle, you can think of it as very similar to the water cycle. It's the motion of carbon around the Earth instead of water, but it's the motion of carbon from the atmosphere, maybe being taken up by plants and put into soils, being weathered and transported down rivers to the ocean, and then also uptake of CO2 by the oceans, release by respiration or fossil fuel burning. So it's the movement of carbon around the Earth system. And so once again, kind of why why study the carbon cycle? Why focus on carbon in soils? And the obvious answer that comes to mind is climate change, right? So rising CO2 in the atmosphere from human-induced burning of fossil fuels increases temperature, changes rainfall, increases storm severity, and causes sea level rise. And so this is a kind of the most prominent idea of why people focus on the carbon cycle. Um, and so here's just a figure showing CO2 emissions from humans and atmospheric CO2 in blue here, showing that they kind of trend together. And so the primary reason for studying the carbon cycle is climate change, but there are other reasons, right? So in general, CO2 in the atmosphere alters the rate of weathering, and this effect of changing weathering rates is one of the main reasons that our planet has stayed habitable over kind of millennial or million year timescales, and hasn't had a runaway greenhouse effect like Venus, let's say. And that's because of as CO2 rises in the atmosphere over these long timescales, weathering processes will slowly take it out of the atmosphere and turn it back into carbonate rocks and deposit at the bottom of the ocean. And so that is another kind of interesting thing that we have learned from studying the carbon cycle. Ocean acidification is a current problem that we face with climate change, but we've also studied it in the past and seen how it affects ocean life and actually has caused some of the mass extinctions in the oceans in the past. Um, and then another reason that we study carbon in general and the carbon cycle is to look at how it affects organisms and growth. And so we can see here that increases in atmospheric CO2 actually increases the rate of plant growth, as you can kind of see through these different concentrations of CO2 versus the amount of growth that the rice plant here has seen. And so those are kind of the major and some other reasons that people choose to study carbon and the carbon cycle. And so, kind of to tie all of these things together, I like to bring up feedback loops. So, as kind of I mentioned, my research focuses on carbon in the critical zone, um, and specifically within soils. And one of the major reasons is to look at how these processes that are going on in soils might affect climate change. And that mechanism for how they might affect climate change is what I'm kind of interested in, and that mechanism is often found in feedback loops, and that's what we're usually looking for or trying to understand is the processes that are going on in these cycles and these different um, areas around Earth to figure out how they might affect each other. And so feedback loops are, I find, a bit of a confusing topic for people, but I like to go through them just to give people an idea of why some of these kind of micro topics are important at a global or larger scale. And so, to give an example, there's two types of feedback loops, positive and negative, and I find the naming scheme of them to be kind of the confusing part. So, to give an example of a negative feedback loop, I'm going to focus on this top left one here. This is a just kind of a staple example. And a negative feedback loop is something that wants to return to a stable position or a stable state. So body temperature is a great example of a negative feedback loop. If you were to go outside right now, it's cold out, you would start to decrease your body temperature. So you would start to shiver, which would warm you back up and try to maintain your 98.6 degree body temperature. If it was summertime, you might go outside, it's hot out, you would start to sweat and your body temperature would increase, or sorry, your body temperature would increase, you would start to sweat, and then your body temperature would decrease, and you'd still try to maintain that 98.6 degree temperature. Well, these same types of processes, these same kind of loops of processes happen at global scales. And so a common example of a negative feedback loop in in the environment or for climate change is the one I've kind of given before, which is weathering processes. And so as CO2 increases in the atmosphere, temperatures rise, chemical weathering increases, and that pulls CO2 out of the atmosphere and then allows it to be turned into calcium carbonate and go to the oceans, reducing the CO2 in the atmosphere, therefore reducing the temperature. And so that is an example of a negative feedback loop in the climate system. An example of a positive feedback loop in the climate system, the one I often think of is ice melt. As temperatures rise, ice melts, and what happens is instead of sunlight coming in and hitting that reflective surface of ice, it hits the water or ground underneath, which warms up more, causing ice to melt faster, causing more ground or water to warm, causing ice to melt faster. And so that's an example of a positive feedback loop. And so when we're studying these systems, whether it's in soils, rivers, or wherever we're looking at, we're often trying to figure out how one aspect of the environment affects other aspects and how they might feed back to each other to see how changes in temperature, moisture, rainfall, storm severity, or frequency might change how that ecosystem or how that system functions in the future. Um, And so that is kind of the end of the background section. I hope I didn't go through that too quickly. If I did, you are welcome to ask me questions at the end. Um, but now we'll get into specifically kind of my research goals and what I study. And so my major research focuses on determining what factors play the dominant role in soil CO2 across agricultural and prairie landscapes. And specifically, I seek to kind of figure out how does temperature, moisture, and plants affect soil respiration. So the CO2 concentration and rate of flux from the soil, how is it affected by those three environmental factors? Second thing that we're going to kind of look at today is what is the role of physical versus biological processes, especially during storm events. And so how do biological processes react, or how does CO2 respond to physical change in the environment during these rain events? And then finally, we'll look a little bit at how prairie and agricultural soils differ in their rate of respiration and their cycling of CO2 and carbon. Okay, so in order to do this research, we need to collect a lot of data. And actually, these sites were built or installed before I started working for the University of Illinois. But I've gotten to see a lot of cool pictures and kind of see how they were installed. And I've gotten to use all the data that has come from these sites. And so we have specifically four sites. Two of them are in Illinois and two of them are in Nebraska. Each state has one agricultural and one prairie site, so we can compare them kind of on more local scales. These sites are called the MERS sites. They're the managed induced reactive zone, management induced reactive zone, and they are monitored for a bunch of different measurements at four depths. Those four depths are 20 centimeters, 60 centimeters, 110, and 180 centimeters. Those depths were chosen because the 20 and 60 are kind of the root zone for the agricultural crops generally, and that 110, 180 are the deeper part of the root zone for those prairie crops. And so we wanted to kind of see those rooting areas for the crops and the prairie grasses. And so within these four depths, we measure a whole bunch of different things, including CO2 concentration, oxygen, volumetric water content, and temperature within the soil. In addition, we also take biweekly samples of soil gases, pore water solutes, so the water concentration or the water in the soil, what the chemistry of that is, and then also rainwater, so we can see what's coming into the soils from rain itself. We also monitor a bunch of other aspects of the atmosphere around these sites. So we monitor total solar radiation, rain amount, wind speed and wind direction, and then also snowpack. And so that's what we measure at these sites. And to kind of give you an idea of what the sites look like when they were being installed, and what all these sensors and measurement devices look like. A lot of them are in these pipes, but these four pipes here, they're put into the side of the soil pit at those four depths, and they contain an oxygen and a CO2 sensor that go back to a data logger, and then we can record that data and use it later. These white things here are actually the moisture and temperature sensors for the soil, and so those are buried in the soil at those depths. We have a bunch of manual samplers, so these are gas cups that are inserted into the side of this pit, and then we have tubes that come to the surface so we can measure, we can pull gases out of the soil at those depths. And then over here we have some soil lysimeters. These create a vacuum in them, and they pull water from the soil into the lysimeter, which we can then sample and figure out what the solutes and what chemistry is going on in the soil itself. These pits, oh, sorry, these pits were all backfilled, kind of in reverse order to try to disturb the environment as little as possible. But we did install, or they did install all of the sensors into the side of the pit, into the undisturbed soil, to try to keep it as undisturbed as possible. And so to kind of give you a little bit of an overview of the exact sites we have, right? I said we have two in Illinois and two in Nebraska. The two sites we have in Illinois are called the Allerton Trust Farm. Um, and so that is owned by the University of Illinois. It's been owned by them since 1946. And it's been continuously farmed since that time and actually even earlier. It's been in a corn soy rotation for the duration of our study. It is tile drained at about a meter underground, and it is both limed and fertilized. The Sangamon River Forest Preserve is our prairie site in Illinois. It's been cultivated, or it was cultivated for over 70 years, but it was restored to prairie back in 2007. And then it's on roughly a 3-year burn schedule. And it was actually just burned this past year. or this past summer. Um, and then for the Nebraska sites, once again, we have two. One is in Prairie and one is in Ag. And these sites are actually much closer to each other. They're about a mile, maybe 2 miles apart. They're in adjacent knolls. You can actually see one from the other site. And so they're both located at the Glacier Creek Preserve. The Prairie site was converted to Prairie back in the 1970s, or back in 1970, and it was planted with five varieties of tall grass prairie species, and then kind of left to its own devices since then. Although it is burned on a 3-year burn cycle, and it too was burned this past summer. So the both sites have been on the same burn cycle since the inception of the study. The Glacier Creek Preserve farm um, was farmed beginning in the 1880s, and it is still farmed today. It's also in a corn soy rotation, and it is not tile drained, nor do they add calcium. I should also mention that both sites were corn this past summer, and they were soy the year before that. And so they have, since the beginning of this study, they've actually luckily been on the same rotation, which is nice. And so now let's get into some data that we actually have gotten out of these sensors. And so here we can see the first 2 years of data from the Nebraska Prairie site. And what we have here is we have CO2 in black, temperature in red, and on this y-axis on the right through time. And what we can see here is that temperature has a very nice seasonal pattern, and so does CO2. It goes up in the summertime and it's low during the wintertime, pretty stably around atmospheric throughout the winter. And so uh, CO2 and temperature seem to match quite well on seasonal timescales, which is kind of well known and has been shown in the past. However, as I mentioned, we take a lot of data points, a lot of measurements, and we do it at hourly timescales, so sub-daily timescales. And through doing that, we've actually been able to figure out some new insights. One of which is that even though CO2 and temperature track very well seasonally, they don't track as well on a sub-daily time step, so an hourly time step. So if we look at this top plot here, we have mean monthly CO2 on the Y versus mean monthly soil temperature, both of them at 20 centimeters, on the X. And we can see these points plot really nice together in kind of a linear fashion, right in a line here. And that's kind of that seasonal pattern that we see in temperature and CO2. They track very well together. But if we don't take the averages and we take instead those hourly samples, we get this bottom graph, right? Hourly CO2 on the Y versus hourly soil temperature at 20 centimeters on the X, on the horizontal part. And we can see that even though there's a pattern, it does increase with temperature. If we were to take like 20 degrees Celsius and look at the range of CO2 we might see, we're anywhere from about 1,000 to almost 10,000 ppm. So we're an order of magnitude that CO2 can range all at the same temperature. And that's true for most temperatures. So although the trend is very well defined, temperature is not actually a great predictor at daily or sub-daily scales for CO2 concentration. That means that there's a lot of other things that are at play beyond just the seasonality of temperature that control the processes that alter CO2 concentrations within the soils. And so one of those things is water. Um, and so rain events and storm events alter CO2 concentrations in soils. And we have here on the right 4 examples of storm events. So the two y-axes, you have the left y-axis is CO2 concentration. The right one is the volumetric water content. It's the wetness of the soil. And through time, you can see that blue line is the water content and that black line is the CO2. And if you were to look pre versus post these 4 storms, you notice a trend where CO2 is lower on average pre-storm and higher on average post-storm. This is pretty well known. It's called the Birch effect. And it is basically the microbial communities with that boost of water start to respire more CO2 or respire more carbon and CO2 faster, and they're more active. And so CO2 concentrations in the soils rise. And so that is kind of part of the reason why in those previous plots we saw that temperature isn't necessarily a great predictor because there's other things that play a role on those sub-daily timescales of what CO2 might be within the growing season within the summer. Now, when looking at these plots, there's something else that I noticed, and I want you to notice as well, and it's most easily seen in this one right here, but it's this drop right at the beginning, right at the onset of the storm. You can also see it in this bottom right-hand plot as well. There's that drop in CO2 right at the beginning of the storm itself. And so if we were to look at this plot, you can kind of see it very well. As water content rises, CO2 falls, and this is kind of counterintuitive. Why might we be seeing that? It's not the Birch effect, and so what might it be caused by? And the title kind of gives it away here. Physical processes is what I have figured out, kind of, or it seems to be the cause of this, are what causing this drop in CO2. So what that is rainwater is very low in carbon concentration, and when it percolates into the soil, it takes in a lot of like a sponge. It takes in a lot of the carbon dioxide in the soil, lowering how much CO2 is in the soil gases because it's being taken up by the soil water. And so this drop right here is that physical uptake of CO2 from the rainwater. And this is, I think, important because of if we were to try to figure out how much this increase in volumetric water content affected the microbial community and affected CO2 production, if we were to do this at greater than hourly timescales, let's just say a point pre and a point post the storm, we would miss all of this CO2 that was lost and then had to be remade up by that microbial community. And we would actually underestimate the production of that microbial community by a little bit if we didn't have these hourly time steps, if our resolution was not as good. And so that is kind of one of the important things of having this hourly resolution. The other thing that we can kind of figure out is what conditions might we actually need this high resolution, or might we see this process, this physical process happen. And specifically, it's likely to happen in the summertime when soil CO2 is elevated because there needs to be a big enough differential between the atmospheric and the soil CO2 concentrations to allow that water to sponge up and draw down that CO2. Second off, we need a substantial increase in volumetric water content because there needs to be enough water to actually uptake that CO2. And finally, we need, I say here, the onset of a storm. We need a source of low CO2 water. So irrigation water from groundwater or even river water probably is actually high in CO2 and wouldn't cause this, whereas rainwater would cause this effect because it's low in CO2. And so that's an example, kind of a physical process that could be important for measuring microbial responses of CO2. So next, I want to talk a bit about plants, and one of the things that we've kind of noticed with our data is the effects that plants have, especially during the growing season, on CO2. And so here, I want you to kind of focus specifically on this red circled 2 here. And what these two are is they're the correlation, how well does CO2 trend with either temperature, is this 0.161, or NDVI, which is a metric of how much plants are growing, so plant growth. And we can see here that it trends with plant growth during the growing season better. So CO2 is better predicted by the amount of plants on the environment, on the surface, than it is by sub-hourly soil temperature at during the growing season. And so this kind of correlation here led me down the path of trying to figure out kind of more about this. And so one of the things I found was that CO2 actually in these soils in the growing season shows a day-night pattern that I think is due to this plant mitigated respiration or plant-induced respiration. And so here what we have is if you look at this top plot, it's the hour of maximum sunlight versus the hour of minimum CO2. And we can see that both of those line up right in kind of the middle of the day. So CO2 is lowest when sunlight is highest. This is kind of counterintuitive from what we'd expect, right? Middle of the day, light, and if you look at this bottom one here, and the red here is actually temperature, light and temperature are highest. And we would expect CO2 to go up more with temperature, but instead it's actually at its lowest when light and temperature are the highest, and it's at its highest at night. This is actually something we see often in streams due to uh, bacteria and really more algae growth. Algae lowers CO2 during the day and raises at night. That's not the process that's happening here in the soils, but it seems to indicate a plant-driven process is altering CO2 at that sub-daily timescale. Kind of another line of evidence that this is likely a plant-driven process is these oscillations are only true in the growing season. So if you look here, the CO2 minimums or maximums don't exist in the wintertime. They only exist during that growing season. And so that is kind of another straw in the hat for the the a plant-mediated CO2 production within the growing season, kind of controlling CO2 concentrations. So another way to look at this is to actually look at the CO2 data through time. And so if you look at this top right-hand one, we have CO2 on the y-axis through time, which is the date on the x-axis. Yellow is hours of sunlight. It's about 6 a.m. to 6 p.m. So it's an estimate of sunlight hours. And white is nighttime. We have our different depths here on the right. And so you can see this 20 centimeters is lowest during the day and highest at night. This is also true for the 180, and the 110 is a little shifted, but still pretty true for its lowest during the day and highest kind of at the end of the day. A little shifted compared to the 20 and 180. And so this is kind of showing that that oscillation is happening in a day-night cycle throughout the growing season. Furthermore, this oscillation isn't due to temperature because of soil temperatures only oscillate at that 20 centimeters, as we can see here in the bottom left. 20 centimeters will oscillate in temperature for day-night cycles, but 60, 110, and 180 are kind of too insulated to have much of a day-night effect in temperature for the soils, even though we see these oscillations deeper. And then finally, as I mentioned, or as I said, I was going to talk a little bit about how, or the differences we see between the prairie soils and the agricultural soils, and the rate at which they are producing and consuming CO2. And so this graph here is showing CO2 on the y-axis, and it's showing kind of through time in minutes on the x-axis. And what these are is we took a chamber and we put it on the surface of soil and we watched as CO2 built up in that chamber to get an idea of how quickly these soils are producing carbon dioxide. And so the prairie is the blue and the ag is this kind of yellowy brown. And we can see that the prairie is producing or fluxing CO2 from the soils to the atmosphere almost 10 times or a lot, it's an order of magnitude more than the agricultural soils. Now, what this means is that CO2 is leaving the soils and going to the atmosphere faster in prairies than agricultural soils, but it does not necessarily mean that prairies produce more CO2. Because of if we look through the kind of same time period, this prairie site has actually increased the amount of CO2 in its soil concentration more so than the ag site. And so what this actually means is that the prairies are cycling carbon faster than the agricultural sites. So they put carbon into the atmosphere and they pull it out of the atmosphere back into the soils at a faster rate, cycling it between soil and atmosphere much quicker than what we see in the agricultural sites, meaning that prairies can respond faster to changes potentially in the carbon cycle than the agricultural sites can. Um, and so, in conclusion, uh, plant root mediated soil respiration drives growing season soil CO2. Temperature seems to be a good seasonal indicator of carbon dioxide, but doesn't do a great job at sub-daily timescales. And then both physical and biological processes are important to consider when understanding the production of CO2 and CO2 concentrations during events. Um, Thank you. I'm happy to take any questions that you might have.
Thank you for that great presentation. Yes, if anyone has questions, again, feel free to unmute yourself and just ask the questions or put them in the chat. I'm seeing the first one in the chat now. Patricia asks, has a similar analysis been done for urban so non-ag and non-prairie environments?
Um, Not that I'm aware of. Um, I've looked at urban sites in terms of actually streams and not soils in the past. But urban soils are often either covered by impermeable surfaces like pavement or buildings. Um, but it would be interesting to look at like urban parks because those are very managed systems that may have kind of a change or potentially even near urban kind of oil or pollutants because of sometimes those actually will spur on microbial um, respiration depending on how bad it is.
Okay, Carrie asks, what are your next research questions you will investigate based on your current conclusions?
So currently, what I'm working on from this stuff actually is trying to understand. We have very high resolution data, but most people don't. So we have 1 hour data for soil CO2, which allowed us to see that those physical processes, those are often not captured. And then also, most studies kind of just assume or see a very strong correlation with temperature because they're looking at much like 2 week or monthly sampling points. And so we can kind of see that there's a lot of other processes that are controlling soil CO2. And so I would like to look more into what those other processes might be. And I've been actually working on updating a soil plant and reactive transport model. So it combines what the plants are doing, the chemistry in the soil, and then the physical diffusion and motion of gases in the soil into discrete different things to try to see where the CO2 that's being respired by these microbes is coming from. Is it coming from the roots? Is it coming from the exudates, the stuff that the roots give off in order to spur on microbial respiration? Is it coming from the decomposed organic matter from last year or from previous years? And so that's one of the major things I'm interested in. The other one is I'm looking at how does the resolution of your data affect your predictions for these soil CO2 fluxes? Um, because if it's been shown in actually by this group in the same group that I work with in rivers and streams that higher resolution data helps to predict uh, CO2 fluxes from rivers much more accurately. Lower resolution data causes over or underestimations of river solutes. And so we're trying to do this a similar thing for soils to see when and where do we need this really expensive but really useful high resolution data so that other researchers can kind of budget and figure out, do I need 1 hour data? Can I get away with half hour or like 1 day data or weekly data? What do we need for our specific research goals? So those are the two things that I'm kind of working on now from this current research.
Okay. Um, so Jennifer asks, do you have any info on herbicide effects on soil microbes and therefore also on carbon dioxide sequestration by microbes?
So I don't know actually if these agricultural farms are using any herbicides. I'd imagine they are. I I I didn't see any clear like drop-offs in soil CO2 throughout the growing season. I'd imagine it's not great for these communities, but I haven't really looked into it too much. I'd be interesting to ask some colleagues maybe about the microbial community diversity in agricultural versus prairie. I'd imagine prairie has a much higher diversity and therefore can kind of withstand changing environments much better than the agricultural sites could.
Okay. Madeline wants to know, do pocket prairies in the urban setting have any influence or are they too small?
So, um, in I would say they have an influence. For this kind of research, I'd imagine they actually act very different. They probably have a lot more pollutants than a native prairie, um, just because of air pollutant, and it's going to get captured by rain and then rain down onto that system. Um, but they small agriculture or small parks, I mean small prairies, small parks in urban areas are very good for water. So that's where a lot of my background before has been in. And you might have seen rain gardens and other kind of installations to capture urban water. and put it into a green space like a park, and having a healthier soil can mitigate a lot of issues with pollutants in rainwater, and kind of take up nitrogen and take up other pollutants and put them into plants or remove them from our waterways. So they do help, but I don't know how much they change the CO2 cycling. They're not very big, so they wouldn't be able to store a ton of carbon.
Okay. Patricia would like to know, could your research be used by industry when carbon credits are being assigned? For example, moving a combustibles industry to a prairie or field area for better results.
So I don't know about moving the actual plant or anything along those lines, but in general, carbon credits are based on this type of research. At one of the places I've worked in the past, Great Mountain Forest, I did a little bit of field work for a researcher that was trying to understand what portion of trees are actually, like what portion of a forest's trees are actually rotted out in the center. Because of if you consider that rotted center as good wood, you are overestimating the amount of carbon that that forest is storing. And so his research was going into kind of trying to make a better model to predict how much CO2 is actually, or how much carbon is actually sequestered in an acre or a hectare of forest. And that's the same kind of that can be used for this. If we can model the rate of CO2 sequestration in these prairie systems and understand when they are and when they're not. So like in these two systems, Illinois hasn't really changed its CO2 concentration, or its carbon concentration in the soil very much over its restoration, whereas Nebraska has. Now there's two things. Nebraska has been in prairie restoration for a lot longer, versus the Illinois site, but also Illinois is a lot wetter, so it might have already maintained or been closer to its peak carbon sequestration prior to this. Time will tell, but this kind of research is what helps the people that are making those carbon markets understand how much that environment is actually worth in carbon.
And Jennifer has posted an article that you might want to look into as far as the impacts of pesticides on soil microbiota. Um, and then Carrie has a question. Would you conjecture that a cover crop used and then seed drilled versus plowing would change the agricultural soil results?
Um, I would think so. So one thing that we notice with the agricultural soils is um, the season seems to be a little bit condensed, and they are a lot more seasonal in their trends, like sharply seasonal in their trends and patterns than the prairie soils. So a cover crop could potentially um, make it so that maybe the early season response is a little faster. Um, and then I would think that the different methods of how you're going to plant and how you're going to till the soil is definitely going to change things. So tilling and aerating the soil is going to kind of potentially spur on some microbial respiration that might almost deplete its soil of carbon. If you continuously till a garden, oftentimes you have to continually add carbon to it, leaves or other organic matter, because of what you're doing is you're allowing those microbes to kind of eat up all of that carbon because they have plenty of oxygen, plenty of that resource, and so they're going to run out of something. It's either nitrogen or carbon, and with how much nitrogen we put on the fields, that probably induces a higher carbon use.
Right. See, that's all the questions we have at this point in the chat. I was kind of wondering, I noticed that both of the prairies that you were looking at are restored prairies. Do you have any inclination as to what might be different with your results if you would be looking at, you know, good to high quality remnant prairies that hadn't maybe ever been plowed, or at least not within recorded history?
I would think they're like, especially in the Illinois one, I would be really like interested to see because I would think there would be some differences. I would think that um, has potentially higher carbon concentrations in the soils. Or it would give us an idea, it would give us the answer of, well, is it just because we haven't had enough time, or is that actually the most that these soils will naturally hold? Um, but that's still like an up in the air question of that. So it would be really interesting, but I don't know of many um, prairies that haven't been tilled at some point. It's pretty hard to find one, especially one that you can then go dig a soil pit in and install a ton of sensors.
Yeah, I imagine they don't like people digging in those if they can help it. Um, so Erica was wondering, what would you consider to be the most important takeaway for a middle school science student who likes to learn about your research?
Um, I mean, so I try to include a lot of background in a lot of my talks, because of I think that oftentimes the like individual presentation or topic that many scientists are presenting on is very niche. It's very like single-minded, tiny. So that's why I like to have that feedback slide in there. And I think that is kind of one of the biggest ones to me is that oftentimes these niche topics are important because they are a building block into a larger system. And that's kind of what that feedbacks is what I'm trying to use that feedback to kind of illustrate is that soil CO2 in a single prairie or agricultural site in Illinois isn't really going to tell us about carbon cycling around the world, but it's a building block that if I and many other researchers look at that kind of stuff, will tell us how these processes work, and then that will allow us to understand the carbon cycle in general. And so I think that's kind of one of the key takeaways I would say. Um, and then other than that, I guess specifically on the research itself, that prairie soils seem to kind of respond faster, right? They take up CO2 and they produce CO2 faster. So they can act and change and cope better than an agricultural soil could.
Right. Very good. Um, I guess one other question I have is knowing that, having been to the Sangamon River Forest Preserve myself, and I know some of our other volunteers have as well, I'm curious if that site has ever been flooded, or at least with the equipment there, and if so, if that kind of gave any interesting insights to your research.
Um, so I don't know of any. It's been a drought for the last 2 years
Okay.
that we've had, or for almost the last 3 years that we've had these sensors in. They were put in 2021, and so they've been 2021, 22, 23, or I think end of 2021, 2022. So they were in during 2022 and 2023, and and then this year, and all three of those years have been pretty dry. So they haven't, I don't think they've been flooded yet. I do know that some of the lower, the 110, 180 sensors do actually, the groundwater will come up and flood those sensors. So some of the CO2 data that we're showing is groundwater concentrations, and it's the CO2 in equilibrium with the water in the soil and not the air in the soil. Um, it would probably change things if it was actually to flood more frequently because of you'd get more anoxic, so you'd deplete the oxygen, and you'd probably see some interesting uh, differences. But I'd imagine that it would still be pretty biologically driven, like what we see.
Okay, wonderful. All right. It's been a lot of people saying thank you, an excellent presentation, and I must agree. Um, yeah, very good information. And yeah, people already saying that they look forward to watching the video of it just to kind of get more insights from this.
Yeah,
Um,
thank you.
so yeah, I'm not seeing any other questions. Thank you for the great presentations, and thank you to all of us for joining us thanks to all of you, excuse me, for joining us tonight. Before you go, I wanted to mention that we are currently scheduling our winter speaker series, and that will occur in January and February. Also, if you're interested in learning more about how road salt use is impacting Illinois streams, we do have our final winter chloride watch training session coming up this Thursday night. You can register for these events and other future activities on our webpage, on our Facebook page. So we look forward to seeing you at our other events in the future. Thank you for joining us tonight and have a wonderful evening.